Okay, I will talk today about neural codes for natural behaviors in flying bats, about our work. By the way, I don't know if you normally ask questions in the middle. That's perfectly fine. If you want to ask questions in the middle, maybe after each section, or if you want to wait to the end, that's also fine. You know, whatever you prefer is okay. But I'll be happy for questions. So before I start, just to tell you that three things as an introduction. One is that our goal, or my goal, is to understand the neural basis of behavior. So I'm interested in the neural basis of behavior, broadly speaking. And of course, you need to focus on something. So I focus on spatial behaviors like navigation, spatial memory, spatial cognition, etc. And also more recently, in recent years, about social behaviors. And the take-home message that I would like to convey today is that it is crucial to study the brain under naturalistic behaviors, or closer to the natural behavior of the animal. And I will illustrate why via several examples of studies from my own labs, which are about different stories. So they might be a bit heterogeneous, but I think the underlying theme is that each of these stories illustrates the power of studying the neural basis of natural behaviors. And I hope to convince you that it's important to go towards more naturalistic or natural behaviors. And the third disclaimer is that we work on bats, and as it is clear from my opening slide, and bats are cool and interesting, but it's important for me to say explicitly that my goal is to understand the brain, not to understand the bats. And bats are not are a means to an end, and the end goal is to understand the brain. But as I'll show you, because they have interesting and somewhat unusual behaviors, we can use bats to ask questions that are difficult to ask in standard animal models like mice or rats, or in humans. But the purpose is to understand the brain and to glean general insight about brain function, about system neuroscience, behavioral neuroscience in a broad sense. Okay, so after having said that, so as I said, I'm interested in the neural basis of behavior. And I often ask myself, which behaviors should we study? So, if you're interested in natural behaviors outdoors, let's say in social behaviors or group behaviors, you might want to study something like this: group hunting in lions and communal defense in buffalo. This is very interesting. These are very interesting behaviors, both by the lions and by the buffalo. But of course, well, first of all, probably the safety unit of my university will not approve such a study so easily. And secondly, you might say this is not such a controlled behavior. It's hard to study in a controlled manner. So most people that are interested in social behaviors are doing something more like this: tube test in mice, where they have two mice pushing each other in the tube, and basically the one who is the winner is considered higher in the hierarchy. So it's a classic test to determine social hierarchy. And so I think a lot of us have study behavioral neuroscience, have in mind this trade-off between, on one hand, controlled laboratory experiments that are controlled but unnatural, and on the other hand, natural behaviors that are natural but uncontrolled. And I'd like to argue today that this is not so simple. So this trade-off is not precise. Why? Well, first of all, this behavior is not exactly uncontrolled. It's not that the lions or the lionesses are running randomly. They're actually performing hunting behaviors in a very relatively stereotypical manner. They have particular strategies for hunting that they repeat over and over again. So it's not exactly uncontrolled. In fact, they are repeating trials of hunting, if you will, in a relatively reproducible manner. So this is not exactly uncontrolled. On the other hand, this is also not exactly unnatural, because mice are burrowing animals. They live in burrows underground, and from one mouse to meet another mouse in a burrow, like in this tube test, is actually quite common outdoors. So it's not exactly unnatural either. So I'd like to argue that this is more of a two-dimensional plane. It's a conceptual plane. Of course, one is artificial versus natural behaviors, and the other is uncontrolled versus controlled behaviors. Now, of course, you can have extreme situations, for example, or extreme cases. For example, the classic head-fixed self-stimulation experiments where a rat or a mouse is pressing a lever to deliver a current in some dopaminergic area in the VTA or the MFB, to release dopamine. Those experiments have been run since the 50s and 60s. These are highly controlled experiments, have been used a lot, but highly artificial. And on the other hand, 24/7 unconstrained, completely natural behavior is very natural, but uncontrolled. So this diagonal line is sort of this axis that I showed on a previous slide. But then you have cases off the diagonal. For example, one of my favorite examples is birdsong. Birdsong in zebra finches, at least, is highly reproducible. The male birds sing again and again, hundreds of times per day, the exact same song with a precision of 1 or 2 or 3 milliseconds. So extremely precise repetition. So if you wish, the birds is doing a control experiment for us. It's a controlled or a reductionist experiment in the sense that the bird is controlling the experiment for us. It's repeating the exact same behavior over and over again, extremely precisely. I mean, you would wish in a mouse to get a precision with a 1 millisecond precision, no matter how much you train it. And here they do it naturally. So it's a natural behavior, but highly controlled. And then maybe the lion hunting is somewhere here, also very natural, but a bit less controlled. And there are many examples like this. Hunting in behaviors in bats, many social behaviors. There are many, many examples that are natural, at least to a large degree, but also controlled to a reasonable degree because the animal is repeating the same behavior over and over. So I would like to argue today that this is the sweet spot or sweet area where we as neuroscientists should try to go to. In the neural basis of navigation that I'm studying, this is the direction that we are going now. Okay, so this is sort of a general introduction about sort of the philosophy of the research in the lab. And now, let's get to to business. So, to introduce you to the neurons that are related to navigation, or sort of a short introduction on the spatial neurons in the brain. I'm sure you all know about this, so I'll do it very briefly. In the hippocampus of rodents, if you stick a tetrode or in the hippocampus of a rat and record from one neuron, you will see something like this. This is one example. This is a rat that's running in a 1 by 1 meter box. And so the black line is the trajectory of the animal. It's not really a navigation task, it's a random foraging task. The experimenters are throwing randomly food pellets for their rats, and the rats are running around randomly. And one neuron in the hippocampus is firing. Here, you can see the firing of one neuron in the hippocampus. In red dots are the spikes of this neuron, or where was the rat when the neurons were fired. So this neuron fired mostly in the northeast corner. You can turn it into a heat map, and you see this neuron fired mostly in the northeast corner. So John O'Keefe discovered these kinds of neurons, called them place cells, and one place cell fires here, another place cell will fire here, a third place cell will fire in the south end. So together, if you look at these neurons as a population, they encode sort of a cognitive map of where am I in space. Then there are grid cells in the entorhinal cortex that the Moses discovered that are, the experiment is identical basically to the experiment with the place cells, an open 1 by 1 meter empty box, random foraging, but these neurons fire not in one location, but in a set of locations that form a hexagonal lattice tiling space. And so these neurons can be thought of perhaps encoding distance traveled. And then there are head direction cells, neurons that don't care so much about where am I in space, but more about the direction. So here's an example of a neuron that fires mostly when an animal looks east. Another neuron might fire when the animal looks north, etc. So this is sort of like a neural compass. So, okay, so we have neural maps in the brain, neural distance meters, and neural compasses in the brain. So these are all the elements, if you'd like, in order to build a navigation system. Now, what we ask in my lab is, how does real-life navigation differ from navigating in small laboratory setups? So you can give several answers. the world is not flat, it is complex and three-dimensional. It's actually two different things, complex and three-dimensional. So how does that affect the system? The world is not 1 meter in size. Navigation occurs over kilometers, right? So how does the scale affect the system? Often animals navigate in groups. So how does the social factors come into play? And also when we navigate, we often switch to other behaviors. We don't just navigate, we also do other things. So how do switches between navigation and other behaviors affect the system? And you can give more answers here to this list, but these are things that we have been studying experimentally in my lab, and this is also the outline of my talk today. These are the things I'll be talking about today. And we ask these questions in bats, as I said already, specifically Egyptian fruit bats, Rosetus aegyptiacus. These are very large bats. They weigh between 150 and 180 grams or so, the males, much bigger than bats that you know from Russia or from Europe or from the United States. They're much bigger than the typical bats in most countries, except the tropical areas. You'll just by looking at their eyes, even if I don't show you any data, you'll believe me that they see very well. In fact, there are 1,300 or 1,350 species of bats, so about almost a quarter of mammalian species are bats. None of them is blind. But these guys in particular see very well. They have visual acuity that's much better than rats or mice. And also they have the famous sense of echolocation, where they emit an echolocation click, and from the returning echoes, they can localize objects in the environments. I'll come back to echolocation a little bit later. But from practical perspective, because they are big, we can put quite a lot of weight on them. So we can put neural logger systems, localization systems, etc. So they can carry quite a lot of electronics. Of course, we also miniaturize the electronics a lot, but it also helps to have a bigger animal. Okay, so just to put you into the mindset of what does it mean to navigate outdoors for a bat like this. This is from a study we've done already some years ago, where this is purely behavioral study, where we did it in these bats outdoors. They're very common in Israel. So this was done in a cave that's about half an hour drive from my lab, halfway between Rehovot and Jerusalem. And we just glued on the backs of these bats these little glued here, these little GPS data loggers. So these are just little devices that store the position of the bat as it flies outdoors. And this is the kind of data that you get, and the behavior that the bats exhibit. This is an example from one bat that was flying from its cave. They roost in caves. It was flying to a particular fruit tree, right? These are fruit bats, so they go to forage every night in fruit trees. But this bat and all other bats basically were flying to a particular fruit tree. Each bat has its own, might have a different tree, but an individual bat goes over and over to the same tree. So these are data from one bat over 7 nights. So each line here is a different trajectory of the same bat over different nights. And it goes really to the same tree. I mean, you can't really see it, but if you zoom in, it's one tree that they go to. And, they fly, in this case, about 15 kilometers to this tree. So these guys can really navigate. They can go over and over to the same tree over this, very straight, over a distance of 15 kilometers. So this is the kind of behavior we'd like to understand eventually. Okay, so let me start with the first point that I've made of how natural navigation is different, that the world is three-dimensional. So I'll talk a little bit about 3D spatial codes in the brain, 3D place cells, 3D direction cells, and 3D grid cells. So first of all, before I go to 3D, let me tell you in 2D, when we ratified the bat, we put the bat in a box and let it crawl in 2 dimensions, just like the rat, the black box or the white cue card, exactly as these experiments are done in rats, so that we can compare. We found basically the same types of neurons, just like in rodents. We found place cells, grid cells, and head direction cells. So generally, in 2 dimensions, it looks very similar to rodents. Then when we went to 3 dimensions, Michael Yartsev, when he was a PhD student in my lab, he recorded from 3D from place cells, hippocampal place cells in flight. We have a large flight room. So this is a 6 by 5 by 3 meter room. So pretty large flight room. And we developed wireless devices to record wirelessly electrophysiology from flying bats. I'll talk about this a little bit more later. And what Michael found is basically shown here. This is one example neuron. The color here is just as before, is the firing rate of the neuron. And you see that this neuron doesn't fire throughout most of the room, but fires in a spherical area in the middle of the room. So whenever the bat flies through a particular sphere in the middle of the room, this neuron fires. And the size of this sphere is 80 or 90 centimeters, something like this. And the fact that it's a sphere, indeed, in the vast majority of the neurons, more than 90% of the neurons, this was a spherical place field, means that the neuron have the same resolution in the X, the Y, and the Z dimension. Then a couple of years later, Arseny Finkelstein, who was also a PhD student in my lab, by the way, today they both have their own labs as PIs. Arseny Finkelstein asked, is there a 3D neural compass in the brain, 3D head direction cells? And again, I'm telling this very briefly because this has been already published. The short answer is that yes, there are 3D head direction cells that respond to a particular three-dimensional direction, there or here, like that. And there was an interesting twist in this study that I don't have time to talk about, that Arseny was able to show that the coordinate system in which the brain represents 3D directions is not the classical spherical coordinate system, which is the textbook mathematical coordinate system to represent directions in 3 dimensions, but rather it's a toroidal coordinate system where there's 360 degrees of azimuth and 360 degrees of pitch. And this solves all sorts of problems like non-commutativity of 3D angles, and the fact that you have discontinuity problems in the zenith and the classical spherical coordinate system. So it solves all sorts of problems, but this was an experimental finding. Again, if anybody will be interested, I can talk about this later today. And just to tell you that following these findings, several groups looked for 3D place cells and 3D head direction cells in other species, and they found them. So 3D head direction cells were found both in monkeys and in mice, and had very similar properties to the ones in bats. So monkeys, of course, are very much two-dimensional animals. Mice are also good climbers. Also 3D place cells were found with delimited place fields like here. So it seems to be a general thing in mammals, not just in bats, which is nice. Now, what about grid cells? Are there 3D grid cells? This is a work by PhD student Gili Ginosar, and I'll tell a little bit in a little bit more detail about this. So in 2 dimensions, here's a beautiful example of grid cells in 2 dimensions from the Moser lab. Grid cells are characterized by 2 main properties. One is their famous hexagonal lattice, and the second is there's a fixed local distance scale, meaning nearby firing fields have a fixed distance between them. Now, you'll immediately tell me it's the same thing. If you have a perfect lattice in 2 dimensions, you'll also have local fixed distance scales, right? And that's true in 2 dimensions, but it turns out that in 3 dimensions, these things are separate. So you need to look at each of these properties separately. So is there a global lattice versus are there local distances in 3 dimensions is not a trivial question, and I'll show you that. So what might we expect to find for 3D grid cells? We might expect to find vertically aligned firing that forms a 2-dimensional lattice, but based on cylinders. We might find one of the perfect sphere packing arrangements like FCC or HCP, face centered cubic, or hexagonal close pack. These are the optimal way to pack spheres in 3D-dimensional space, at least infinite space, and this has been hypothesized by several studies that sort of this is what we should find, because this is the 3-dimensional generalization of the hexagons that the Moses found in 2 dimensions. But of course, we might find something else, like other types of lattices, and we might also find a disorder arrangement, or maybe something in the middle between order and disorder, something like a local order. So these are the types of representations that we wanted to consider. And so to look at that, we recorded from the medial entorhinal cortex of flying bats in the big flight room that we have, as I said, almost 6 by 5 by 3 meter room. Gili put these landing balls for the bats with food on them, very close to the walls around the perimeter of the room. And there were these landing balls were at various heights. So the idea was basically to encourage the bat to fly very much in 3 dimensions. So to fill the 3-dimensional volume of the room, which they did. Here's an example of one recording session. They flew very nicely and filled the 3-dimensional volume of the room, and the distance flown per session was typically almost 2 kilometers per session. So they filled the volume of this room quite extensively. And here are examples of 8 neurons that Gili recorded in the entorhinal cortex. And already by looking at that, you can see that there are fields here. So there are like areas of activation, many of them, not one as I showed you in the hippocampus, but many fields here and here and here. You also immediately see that there is no vertical elongation. It doesn't look like there are columns here, so we can rule that out. We also did specific analysis, explicit analysis to show that, but I won't show you that. Just by looking at that, you'll believe me that there are no columns here. We verified this. So then, to understand what do we see here, first of all, we looked at these local distance scale. Is there a local distance scale? So we identified these firing peaks, these grid fields, as you see here. And then we basically asked, we quantified the distance of each grid field, of each firing field from its nearest neighbors. So we looked at this field and its distance from its 3 nearest neighbors, and then the next field, the distance from its 3 nearest neighbors, and we collected those local distances, of course, not counting twice. And then we can construct a histogram. For example, for this neuron, this is the histogram of these local distances. And you can see that the interfield distances, histogram is very narrow. The distribution, this histogram is distributed narrowly, meaning that it looks like there is a local distance scale that is conserved. And to quantify this, we looked at the coefficient of variation, the standard deviation divided by the mean of this histogram. So for this particular neuron, the coefficient of variation is 0.17. So that's a low number. It means the histogram is narrow, but more importantly, it was lower. The CV, the coefficient of variation, was lower than for 1,000 shuffles. So that means that it's a significantly narrowly distributed. In fact, for 20 to 30% of the multifield neurons in the medial entorhinal cortex, we found this local distance scale. So these are grid cells. Next, we asked, is there a hexagonal lattice, this 3D lattice that is a 3-dimensional generalization of the 2D grids found in on 2D surfaces? And the short answer is no. Okay, it's hard to show a negative result because we worked for 2-3 years trying all sorts of analysis, and the short answer is that nothing worked. We could not find any hints for any 3-dimensional global lattices like an FCC or HCP lattice that fills space. So we can quantify. We did several analyses here. For example, this is an example neuron where we quantified we looked at the fit error, so the distance, average distance from the fields, the actual fields of the neuron from the best fitting FCC or HCP lattice, where we just in a brute force manner did all possible combinations of FCCs and HCPs with different orientations, shifts, and scales, 3 million combinations. And so the best fitting fit error in centimeters is not, or in meters, sorry, is not better than for 1,000 shuffles. So this neuron is not an FCC or HCP neuron. In fact, none of the neurons. We had 0 neurons that showed an FCC or HCP lattice. We also looked at distributions of angles. Again, I don't have time to go into the details here, but we'll look at, are there like angles at 60 degrees or at 90 degrees or any other prominent angles when you look at local angles that will hint at some lattice? And the short answer is no, we did not find any prominent angles. And so we can conclude that basically we do not see this. We don't see a lattice because we FCC or HCP because we explicitly tested for that, but we don't see any other lattices as well because we looked at the angles. On the other hand, this is not a disordered system because we do see those local distances that are preserved, local distances between nearby firing fields. So it looks like we have a situation of a locally ordered system. So to understand this, what does it mean and how can it come about? We teamed with 3 theoreticians, Yonatan Al-Khadef, Yoram Burak, and Chaim Sobolinski, trying to model these results and trying to understand the difference between what happens in 3 dimensions versus 2 dimensions within one model. So the idea that in the next few slides, when I'm going to say we, it's actually, they did the work, but, it's also in the published paper. We had the model results as part of the experimental paper. So basically their idea was to look at local interactions. In a lot of physical systems, systems in physics or in chemistry, you can model them by local interactions between particles, and then the system can be either in a disordered state or in a very ordered state or in something in between, depending on the parameters of the system and on the noise. So basically, we wanted to understand whether this kind of a pairwise interactions, where now we'll treat the firing fields as particles, can this explain what we see, which is something like this here in the bottom, and not an ordered lattice. Okay, so specifically, we use the Lennard-Jones potential, which is this potential here, which is a classical potential used in physics and chemistry. As I said, this is how it looks. So this is potential energy versus distance between the particles, and it has an area of repulsion. So, the derivative is negative at short distances. It has attraction at long distances, and it has a minimum that the system, minimal energy, where the system will normally converge, except that you have noise or a temperature that would introduce fluctuations, and the field position or the particle position will deviate from where they're supposed to be. And it turns out that such a potential or a similar type of a potential can be achieved in a self-organizing neural network with competitive plasticity, because the question that we're often asked is, this is a phenomenological model, but can you realize it in a more mechanistic neural network? So the answer is yes. This model explained the data much better than random Poisson arrangement of the field. So just to show you, the y-axis is the distance, is some distance metric between the data and the model. So the lower the distance, the better is the model. And so the pairwise interaction model, this Lennard-Jones model, was much better than in a random arrangement of fields. So that's another way to say that the fields were not arranged randomly. In fact, there was an order, just that it was local. We also compared to a jittered FCC or jittered HCP, because one of the ideas that you might think about, maybe you do have an underlying perfect lattice, but then individual fields deviate randomly from the perfect lattice position. So we compare to that kind of model, and again, the pairwise interaction model explains the data much better than jittered FCC or jittered HCP. So basically, we conclude that this model does a very good job. We also compared to a host of other models I don't have time to go into, it's all in the paper. And then we wanted to ask the cardinal question about 2D versus 3D. How can we understand this difference between 2D and 3D? And to compare that, we use the radial density function. So the idea is that if you look at this perfect lattice here on the left, so if you look at a particular field, and so when you go to a distance 1, so to the nearest fields or nearest particles, you'll get a peak in the distribution. The radial distribution function is basically a function measuring the distance, all possible distances between particles in a system. And then when you go a bit further, you'll get a second peak, because again, you have many particles at this distance, etc. So you'll get multiple peaks at distances 1, 2, 3, etc. But when we did 3D simulations of the model, we did not get peak like that. We got something without a peak. On the other hand, when we simulated the 2-dimensional room, so we used the exact same model, the exact same equations, just we took the 3-dimensional room and we squished it to a 10-centimeter height. Okay, so now it's a quasi two-dimensional system, but it's really three-dimensional, so we can use the exact same equations. And here we got this first peak, second peak, third peak, etc. So we do get these multiple peaks. And in fact, when we looked at individual realizations of the simulations, we found many, many examples. Actually, the large majority of neurons were arranged in some sort of hexagonal lattice, as you can see here. So in 2 dimensions, this model gives the 2-dimensional hexagonal grid structure, but in 3 dimensions, it gives something like what we see in the data, which is the local but not global lattice. And to give you an intuition of what's going on, what's the difference, the basic idea was already known by the physicists a while ago. It turns out that there is a fundamental difference between 2 dimensions and 3 dimensions. So the intuition goes as follows: if you take spheres, so a lot of balls, and you try to put them on a 2-dimensional surface and you squeeze them together, you'll always get a hexagonal arrangement. That's like the best way to squeeze them together. Anybody here who played pool or billiard knows what I'm talking about. You put balls on a billiard table and you squeeze them together, you'll always get hexagons. On the other hand, when you take these spheres or balls and you throw them into a 3-dimensional box, like in a pool of balls in the gymboree, it'll never be arranged in a lattice, because if you just throw a lot of balls into a 3-dimensional box and you shake it, it'll always be arranged in some non-lattice order, because there are many, many possible configurations, and the likelihood of you getting exactly perfect lattice is infinitesimally small. On the other hand, if you look at local distances, like for example, from this ball to its nearest neighbors, the local distances are fixed. In fact, they're exactly equal to the diameter of the ball, because all these balls are jammed, so they're touching each other. And so the local distances are fixed, but there is no global lattice here. And this is basically what we are finding with the 3D grid cells. So this is basically the intuition. The mathematical intuition is that when you have in 2 dimensions, when you have two balls, you have only, or 2 circles, you have only one place to put the third circle. So you have no degrees of freedom. That's why it has to grow to this perfect 2-dimensional lattice. In 3 dimensions, if you have 3 balls, you can either put the next ball here or one phase over. So you have 2 options. You have 1 more degree of freedom. You have a degree of freedom. You have an option. So that already means that it might be here or here, and then the next layer might be there or there. So it's very quickly diverges to a combinatorial explosion. So that's why you don't have a perfect lattice in 3D, but it looks something like that. This is sort of the intuition. So to summarize this part on 3D grid cells, we found cells with multiple 3D fields. 20 or 30% of them are grid cells. By the way, 20 or 30%, it depends on how you do the shuffling. We did it several ways, so that's why the range here is there is a range here. So there are grid cells with local distance scale, but no global lattice. We found 0 neurons that showed a global lattice. And we suggest that the fundamental property of grid cells is not the famous hexagonal structure per se, but actually having multiple fields with a characteristic pairwise distance. And in 2D, this model automatically yields a global hexagonal lattice. So if you confine the model to a 2-dimensional surface, you'll get automatically a hexagonal lattice, as is seen experimentally in 2 dimensions in both rodents and bats. On the other hand, in 3D, the exact same model yields only local structure, these local distances, or a local metric for space, if you will, but no global lattice, as in our 3-dimensional data. And let me just say that this lack of a global lattice in 3D grid cells was also found in rats. So in parallel to us, the lab of Kate Jeffrey published a paper showing no 3D lattice in rats as well. So again, it's not just in bats. It seems to be a general property in mammals. And this has important implication, because the notion that there is a local metric of space in 3D, it argues against models of grid cells that rely on precisely periodic firing, right? A lot of models of grid cells, actually most models of grid cells, both mechanistic models that try to understand how grid cells are formed, and also functional models that try to understand how grid cells are used, what are they good for. Both mechanistic and functional types of models usually rely heavily on having a precisely periodic firing. And if there is no precisely periodic firing in 3D, this is a problem for most of these models. And also this, I think that grid cells, this means that grid cells may underlie maybe short-range 3D navigation, like local measuring distances locally, but certainly not long-distance navigation, or at least not based on the models that require this precisely periodic firing. Okay, I'm ready to move to the next story, but let me know if you want to ask questions now, or should I wait to the end and continue? What's the policy?
We have a question.
Okay, yeah.
Thank you for impressive first part of your talk. My question is about, do you suppose some physiological reasons of such pairwise interaction of grid cells?
Right. So, as I said, it's Yoram Burak. There have been several studies that try to model grid cells by competitive plasticity, so by sort of feed-forward processes, and these create multiple bumps of activity that effectively have interactions between. Now, these are not physiological interactions, but these are effective interactions that are similar in shape to Lennard-Jones. By the way, you don't have to have Lennard-Jones. We tried other types of potentials. They all worked reasonably well. So basically anything that has like a minimum will work, right? And so it turns out that in these neural networks with competitive plasticity, you have effective interactions that act like Lennard-Jones. It's not a real synaptic interaction or something like this, right? So so that's, I guess, my answer. I hope this answers your question.
May I also have a question? The distance of these fields in the pairwise interaction, and first, do they have variability in the longitudinal axis of the entorhinal cortex? And second, do you think, since you suggest that they might underlie short-range navigation, that the scale of these distances somehow is complementary to the scale which can be covered by the cells, grid cells in 3D, in navigation?
Okay, so it's two excellent questions. So one, the first part that you asked about changes along the longitudinal, so the dorsoventral axis of medial entorhinal cortex. You know, for those who don't know, the reason for this question is that in rodents, it has been shown that the scale of grid cells, so the wavelength of the grid, increases as you go dorsoventrally in the entorhinal cortex. In fact, there are some discrete jumps there in the scale. So we looked at that, and the answer is yes. We do see an increase in the scale. Again, the scale in our case is sort of the average distance between nearby fields, but we definitely see an increase in the scale as we go dorsoventrally in the entorhinal cortex. So that's, again, very similar to what has been shown in rodents. And you know, I didn't have time to show it, but it's in the paper. Regarding your second question about long distance navigation, so I don't know, this relates actually to my next part. You know, we never tested grid cells in environments bigger than 5 by 6 meters. So I don't know how they will look, but it's an interesting question how they will look, you know, in an environment that's hundreds of meters in size. So far, we only studied place cells in this environment. I'll show you this in a second. So yeah, I don't know what will be the answer in large scale, but I think it's an important future question to ask.
Thank you. Perhaps we can continue. There is one more question, short one.
Thank you very much for an amazing presentation. I have a question regarding Mikhail Yartsev's paper on place cells in the room with no supervisors. Where he showed that place cells vary depending on the lighting situation. Do grid cells structure vary depending on whether it's dark in the room or we have a light? Thank you.
Okay, so wait, you mean the paper that from my lab, or the new paper from Mikhail Yartsev's lab? I'm not sure.
The April's
okay.
April the 7th of 22.
Right. So with the imaging.
Yeah.
Okay. So, okay, first of all, my answer about grid cells is I don't know. We've never tested grid cells in light versus dark. We did have a study by PhD student Maya Geva-Sagiv, and it was published in 2015 in Nature Neuroscience, where we tested place cells in light versus dark. And I'll get to the new paper in a second. There, what we showed is that there we really tried to understand, do place cells fire differently under two different sensory systems, because they have two distal senses, vision and echolocation, which they can use to probe space from a distance, which is a bit unusual. In rodents, you have one distal sense, which is vision. And then the other senses, like olfaction and whisking, are proximal. They have to get close to something. So here we have two distal senses from a distance, vision and echolocation. So that's a nice opportunity to look at that. So what we did in that experiment, we had bats flying like in a room and on a linear flight track, and we either did experiment in complete darkness, so they had to use only echolocation, or we put lights on, so they used vision, but we played back a strong white noise that blocked their ability of using echolocation. In fact, we showed that if you turn off the light and block the noise, they crash, they can't fly. So this blocking works. So basically, we dissociated here between vision and echolocation. Also, could be odors could be used here, right? Olfaction. So we put ventilators to blow away any odors in midair, which might interfere here. So we worked really hard in that experiment to try to dissociate between vision versus echolocation. So between the two different sensory systems. And what we found is that in the vision session versus the echolocation session, the place cells remap, they fire differently. Even though the behavior was completely stereotypic, they fired exactly in the same trajectory, same speed, overlapping trajectory, everything was the same. Now in Michael Yartsev, a new paper, they did imaging, and they looked at several things. So the stability over many days, which I'm not sure I buy this story, because if you look at their own figures, you actually do see a reduction in the firing, in the correlation between place field over days. So I think they actually, in their own data, do see reduction over days, but okay, this is a separate discussion. With regard to the light versus dark, they actually did not do this. With regard to vision versus echolocation, they did not do this experiment, because what they did is either an experiment in the light, and then the bat used vision, echolocation, and olfaction together that are being used, or they turned off the light. They never had a noise broadcasted. They didn't have ventilators to blow away any odor trails. So in this condition, they used echolocation and olfaction. So no vision, but yes, echolocation and olfaction. So two of the three systems, okay, they did in the second condition, the second session, they don't have vision, but they do have echolocation and olfaction, the same in both sessions. And what they found is that there was actually no remapping, which is different than what we found, but I just think it's a completely different experiment. We really dissociated, have one session where we have pure vision and only pure vision and only pure echolocation, and then we see a difference. And in their experiment, there was a commonality in both sessions, the use of echolocation and olfaction, and they can use echolocation to form the exact same map. So I don't think there's any real difference between the two results. It's just very different experiments. It's not the same experiment that gives different results. It's just two very different experiments. I hope that answers your question.
Thank you. Let's continue, I think, with the lecture.
Okay. So the second part or the second difference that I mentioned for between navigation in the lab experiments versus the real world is the issue of scale. And so this is about representation of very large spaces in the brain, work by PhD student Amir Eliav, who just graduated together with PhD student Shir Maimon. And so bats navigate hundreds of meters underground and also outdoors, as I showed you before. And just to give you the intuition for like what are we going to look for here, or what is the problem? If you look at this example of this bat that I already showed you before, it's flying from the cave to the tree, and the volume that it's covering is huge. The volume in this particular case is 15 kilometers long. It's like a cylinder that's 15 kilometers long by 2 kilometers wide, approximately, by half a kilometer high. How do I know it's half a kilometer? Because with the GPS, we measure that the bats fly between an altitude of 100 meters to an altitude of 600 meters approximately. Okay, so it's 15 kilometers by 2 kilometers by half a kilometer. This is the volume we're talking about. It's a huge volume. And if you want to tile this volume with your small laboratory place fields that you measure in small 1 meter boxes, these fields are typically 10 centimeters in size, right? So if you want to use 10 centimeter spheres to tile this huge volume, you can, make a calculation yourself on the back of the envelope or in your head. And depending on how much overlap you assume, etc., you need something like between 10 to the 13 and 10 to the 15 neurons to have this tiling. And the entire number of neurons in dorsal hippocampal area CA1 in bats and rats is approximately 100,000. So maybe double that, but on the order of 10 to the 5. Okay, so there's a gap of 10 orders of magnitude between 10 to the 15 and 10 to the 5. That's a huge gap. It's not a small problem. It's a problem of 10 billion, right? So it's an enormous problem. It means that there's no way that what we find in small laboratory boxes will translate one-to-one to the real world outdoors. There has to be something else, either scaling up of the place field or something else. And so to understand what is this something else, we turn to an experimental investigation of this problem. So for this, we had to solve 3 technical problems. One is to be able to record on the bat at unlimited distances. So before, in the early studies of the 3D place cells, we used a wireless telemetry system where the data was transmitted locally. So that can work in the lab over a distance of a few meters or tens of meters. That will not work over long distances of hundreds of meters or more. So for that, we developed a neural logger device that stores the data on board. We developed it together with Jacob Wecht, a very good engineer in Israel. And so here is how it looks. So this allows to record basically neural data wirelessly at unlimited distances effectively. And here are some spikes that were recorded in flight. So these 4 channels are the 4 channels of one tetrode recorded in flight from the hippocampus. So this is supposed to, those of you who have seen electrophysiology, it's supposed to impress you that you can get really nice SNR in flight. It works really great. And it's not just electrophysiology. So we have 64 neural channels, but we also have on board an ultrasonic microphone, so we can record the acoustic behavior of the bat, both echolocation and social communication. Also a 9-axis motion sensor. So a 3-axis accelerometer, a gyro, and a magnetometer. We also have now added an altimeter. You can add potentially cameras on board. So you can basically, unlike the classical experiment in behavioral neuroscience where all the behavioral sensors are external to the animal, here we put everything on the animal and let it fly unlimited distances. So this is an entire onboard flying electrophysiology lab, essentially. And we've been miniaturizing this over the years. So this is the smallest system of its kind in the world. And today, the 64-channel neural system that we are using is much smaller in weight than the 16-channel system. This is the old 16-channel system. What we have now is much smaller than the 16-channel system that we had several years ago. Okay, so this is was miniaturized dramatically over the years, and we might switch to 128 or 256 channel system soon. The second problem that we had to solve is how do we measure the position of the bat precisely? So GPS is not good enough because GPS has an accuracy of a few meters. But if we want to dissociate between 10-centimeter place field or something else, we need to be able to measure the position of the bat very precisely. Now, the problem of why GPS is not so precise is that you have the way GPS works. You measure the time differences to satellites in outer space, and in the middle there's atmosphere. Atmosphere is great for breathing, but it's not so great for GPS. There's atmospheric aberration, so it limits the accuracy. So we found this solution from a French company that's doing something very similar to GPS, but locally. So you can put local ground-based antennas. So we deployed ground-based antennas that you see here. And then you measure essentially the distance from a small tag, a really small tag on the bat, and each of these antennas. So each of these distance, particular distance defines a sphere. Here it's shown as a circle, but really it's a 3-dimensional sphere. And then you can cut all these circles or all these spheres, and this gives you the three-dimensional position of the animal. And we calibrated this and showed that this gives about 10 centimeter precision. In fact, sorry, a few months ago, we deployed 40 antennas, and we now have improved the resolution of the system to about 5 centimeter or even 4.5 centimeters over a scale of many hundreds of meters. So we have now a very precise way to measure the position of the bat. And we also need a large but controlled behavioral setup. So we built this tunnel. This is how it looks from the inside. This is the old tunnel. We have now a bigger one. It was 200 meter long, and it's much longer than you see here. Here you see only the end of it, and we put various landmarks in the tunnel. And we let bats fly in there. So here's an example of one session from one bat that was flying. Basically, this is the time in the session. This is the position, 200 meters. And we put food at both ends. And the bats, some of the bats ate the food. Some of them didn't even care so much about the food. They really just love flying back and forth. And so here's the trajectory of the bat flying back and forth and back and forth and back and forth and back and forth. Some of the bats are flying 100 or even 120 laps per session. So crazy. That's 20 or 25 kilometers in one recording session. These are crazy distances. You never get something like that, in a small laboratory environment. So they really fly a lot there. And if you look at the cross section of the tunnel, they really flew in the center. So almost no deviation in the y-axis. They love to fly in the center of things, and also not much deviation in the z-axis. So we turned this into a one-dimensional problem on purpose, because here our question is about the effect of scale. Okay, so we have here a one-dimensional behavioral situation. The velocity of the bats was very fixed. So here is the speed of the bat velocity along the entire session, and you see it's 7 or 8 meters per second, and it's very, very stable along the entire tunnel. This will be important in a second. And so here we're utilizing the bat's fast flight speed in order to measure the representation of large spaces, right? Because we have an animal that moves fast here, we can ask questions about large spaces. If you put a rat here and let it run, it might run a couple of times and then, okay, I had enough for today. So you have to have a fast moving animal to be able to ask question of how a large space is represented and have many repeats for good statistics, right? So here it's like for the question of why bats, here's an example of why a fast flying animal like a bat is useful for studying large representation of large spaces. And so here's an example of one neuron that Tamir recorded in this tunnel. And so all of this is one neuron. At the top, you see the firing rate maps or PSTHs as a function of position, and the bottom are rasters. Each line here is one flight, dots are spikes, so standard rasters, but we've separated them into flight to the east in blue and a flight to the west in red. And what you see here are several things. First of all, there's a difference between the map, the red map and the blue map, or the red raster and the blue raster. So there's a difference between the two flight directions. That's actually old news. We know this already in small laboratory environments, in rodents, and by the way, also in bats in the study that I mentioned from 2015. We also show this directionality there, so that's not new. What is new are three things. One is that the place fields, some of them are really large. Like look at this place field, it's about 20 meters in size, and we had fields that are even 30 or 35 meters in size. So there's definitely scaling up. You increase the environment size, and the size of place fields increases. But the second thing is that there's not one place field. Our classical conceptualization of place fields of place cells, that they have this one Gaussian place field in one location, right? This is like the classical thinking, but here you have many place fields here and here and here and here. And the third is that there is what we call the multi-scale property. So the same neuron can have different sizes of place fields or different spatial scales in different locations. So this neuron has a large field here, a medium field here, and a small field there. This is another neuron that has a large field here, a small field here, a small field here, and a medium field there, right? So this means that the same neuron has different scales or different resolutions in different locations. This is very unusual and very different than what you find in place cells recorded in small laboratory setups. By the way, when we did the same experiment in a small environment, basically we took the tunnel and we put a block, and then we looked at a 6-meter piece. Then we find the same finding as in rodents, namely mostly single fields, right? But when you increase environment size, you find many place fields, and they are different sizes. And there were examples. This is the population. So if you look at the number of cells with various numbers of fields, you see that there are many neurons with 10 place fields per direction, or even 20 place fields per direction. So certainly not one Gaussian, but many fields per direction, per flight direction. And if you look at this multi-scale property, so this is the field size ratio, the largest divided by the smallest field size for the same neuron. So we see ratios of 5, 10, and even 20. 20 means that a particular neuron might have a 1 meter place field here and a 20 meter place field over there, right? So very different resolutions in different locations. And we had control for many, many things. For example, you might wonder, maybe small place fields cluster near landmarks, and this is what creates this multi-field finding. So the answer is no, there's no clustering near the landmarks. We ruled that out. Flight speed cannot explain the results, because flight speed, in fact, was stable along the entire tunnel. So it's not like when I'm flying fast, the field is large or whatever. It cannot explain, and we also explicitly showed that this cannot explain the finding. You very carefully controlled for effects of spike sorting, field definitions, and many, many other things. So I won't go into the details. It's all in the supplementary of this paper. And the short answer is this: multi-scale coding is a genuine phenomenon. Okay, so we have a genuine, very, very different result than found in small environments. So to understand this, we teamed with, again, with Yonatan Al-Khadef and with Misha Tzodix, a theoretician and my colleague from the department, from our department at the Weizmann Institute, and we try to understand what is going on, or what is the function of this multi-scale, multi-field coding. And they did all sorts of things, Yonatan and Misha, but I'll just show you one slide summarizing some of the results. So basically, the idea, the approach was to take a decoding approach, so to compare various encoding schemes. So here on the left is the classical place code. So this is 200 meters, and here are 10 neurons. So this neuron has a small field here, this neuron has a small field here, the third neuron has a small field there, right? So different neurons have small fields in different random locations. And then on the right, scheme 6 is what we find in the bat, which is the same. One neuron has many fields, and they have different sizes. And then we had also other schemes, for example, many fields, but with the same scale per field, per neuron, etc. So we compared various encoding schemes. In fact, not just 6, we had 8 different encoding schemes in the paper, but okay, I'm showing you just 6 here. And what I'm showing you here is what we call the catastrophic decoding error, which is the probability of having a decoding error which is larger than 5% of the environment size. So we did here maximum likelihood Bayesian decoding. We also looked at population vectors, so other decoders, but they all give the same result. And why do we call it a catastrophic error? Because this is a decoding error, a large decoding error, so more than 5% of the environment size, meaning the actual simulated bat is here, but the decoder says it's there. So if there's a large error, we say it's catastrophic, it's bad. And so for small environments, there was no difference. All these encoding schemes gave the same performance, but for large environment, 200 meters, there was a big difference. Note that the y-axis here is logarithmic. So the multi-scale scheme 6, which we found, which is this one, what we see in the bat, was almost 3 orders of magnitude better in terms of catastrophic decoding error as composed to the red line, which is the classical place coding with single small fields. Okay, so there's an enormous difference in the encoding performance for large, but not for small environments. And conversely, this means that with this encoding scheme, you can encode environments up to 1 kilometer with only 50 neurons. But for the red type of coding, the classical coding, you need many more neurons. Okay, so to summarize this part, our presentation of very large spaces, we've seen that spatial coding of large spaces is very different from lab setups. We had many fields per neuron, often more than 10 fields in each direction. There were very large fields, up to 30 meters or even more, but also very small fields, by the way. We had some fields that were as small as 50 centimeters. That's extremely precise, because if you think about it, that animal flies at almost 10 meters per second. So a 50 centimeter precision at 10 meters per second means 50 millisecond precision. That's actually pretty precise. This is not a sensory area. This is a high level area, many synapses from the periphery. So these neurons can be very precise, up to 50 centimeters, but also the same neuron could have a 30-meter field elsewhere. And we showed that this multi-scale coding, as I showed you, the multi-field, multi-scale coding was independent of experience. I didn't have time to show you this, but basically, Shir Maimon, what she looked at, she recorded separate sets of bats from day one. What I showed you so far was from wild-born bats. Actually, everything I'm showing you today, both the 3D grid cells, the large scale, what I'm going to show you in the next stories, is all from wild-born bats, bats that were born in the wild and grew up as adults in the wild and were caught outdoors. I caught them myself outdoors as adults. Okay, so they have definitely seen large spaces and 3 dimensions and everything. But then we also have a breeding colony in the institute, so we can ask about lab-born bats. Do they show the same type of coding, even though they never seen a large environment in their life? And I was sure we will see a difference, but to my shock, actually, there was no difference. Okay, I'm not showing you this, it's in the paper, but the coding, if you look at the histogram of the size of the fields and the number of the fields and the largest to smallest field ratio, it was identical in lab-born and wild-born bats. And also it was there, this coding was there already from day one in the tunnel. So this was really a very strong negative result, if you will, but it really illustrates that this type of multi-scale coding is apparently something very robust and important and fundamental for the system. And we have this theoretical analysis that explains why this multi-scale coding is better for large environments. So it's not just a experimental finding, but we have an understanding of why it's good. And we are now been recording neuron populations. So what I showed you so far was in single neurons, basically, but we've been recording, in fact, neuron populations because we have 16 tetraodes, 64 channels, so we can record neuron populations. And so using these ensemble neuron recordings, we can decode the bat's position in the 200-meter tunnel with extremely high precision. So let me show you. We've used this clusterless decoding method developed by Lauren Frank's lab. I won't go into the detail, but it's amazing. It works really beautifully. So here you see examples of 2 flights where the bat, this is in a segment of 130 meters of the tunnel. The bat flew over 15 seconds. The black line is the flight of the bat, position versus time. And the red line, I hope you can see it, is the decoded position. So it's like overlaps amazingly the data. And here's another example. So really the decoder, it works amazingly well. This is the confusion matrix of predicted position versus real position. And you see that the color is the probability and everything here falls under the diagonal, meaning the decoder works really well. And we had a median decoding error of 50 centimeter in a tunnel that's up to about 200 meter long. So it's 0.25% precision. It's really works beautifully. Now, why is that interesting? Because now we can ask what happens when the animal is in rest. And in sleep and rest, as I'm sure many of you know, you have sequential replay of behavior. People have shown this in rodents where, if you have during behavior, if you look at 10 place cells and order them by the position of their place fields, so during when the animal runs, they're activated in a sequence because you've ordered them this way, so you created a sequence. But it turns out that when the animal is resting or sleeping, the same 10 neurons will replay in the same order, or maybe the reverse order, but generally in the same order in a compressed manner, so about 10 times faster than the replay, than their activity during behavior. So this phenomenon has been termed replay, and this has been studied a lot in rodents, and there are many theories. For example, the idea of sleep, that during sleep, the hippocampus sends these replay events to the neocortex, to the prefrontal cortex in particular, for long-term memory storage. So this is a mechanism of sending information from the hippocampus to the neocortex for long-term memory storage. So there's been a lot of work on this type of phenomenon, and we wanted to ask, in this very large environment, can we find replay? And the answer is yes. There are 3 examples of replay. So this is now position versus time. The color is the probability, right? So this is now the probability of the decoder. And you see that you have here these sequential replays of activity where the decoded position progresses in a linear manner versus time, but on a very fast time scale, right? This is 200 milliseconds or 300 milliseconds. So it lasts for a few hundred milliseconds, and you see these replay events. You'll notice that they don't go through the entire tunnel. They go through a piece of the tunnel, maybe 20 meters or something like that. And they tend to occur during shockwave ripples, for those of you who know what that is. So that's as in rodent hippocampus, they occur during these shockwave ripple events in the LFP. Now, this is the surprising result. If we look now at both during sleep and during awake replay, we'll look at the duration of these replay events. This is just like in rodents, they last a few hundred milliseconds. If we look at the compression ratio, which is the ratio between the replay speed, right? Because here, this defines the speed, right? Position divided by time defines the speed, right? So the slope here is the speed. So you can ask like how fast this replay moves. So you can basically, the compression ratio is if you look at the replay speed divided by the movement speed of the animal during the actual behavior. The distribution of that is quite broad, but the average is about 10. And that's actually very similar to what you find in rodents. If you look at the distribution from rodents, it looks very similar, and also the average is about 10. What is this different is that the distance of the replay is not 200 meters, but it's actually very short, is about 10 meters on average, right? Rarely 20 meters. So these examples that I've showed you, where the replay is 20 meters, is actually a bit unusual, right? 20 meters is a bit of an outlier. Most of the replays are even shorter than that. So you have like snippets of long experience. Now, of course, why is that interesting? Because there was clearly a problem here. If you think about this flight outdoors where the bat flies for half an hour from the cave to the tree, then if it needs to replay this entire half an hour, then if it replays the whole thing with the same compression ratio of 10, it means it needs to sit there and for 3 minutes, 1/10 of 30 minutes, to replay the thing. That doesn't happen. We don't see replays that last 3 minutes. They last short amounts of time. On the other hand, if your replay is fast and you want to replay the entire 15-kilometer flight, then you need your replay speed, within a few hundred milliseconds, then you need your replay speed to be enormous, hundreds or thousands of kilometers per hour. So you need the compression ratio to be hundreds or thousands. You need a very high compression ratio, but that's not what we see. We see actually a compression ratio that is very similar to rodents. So there's apparently some biophysical limitation or some other network level limitation that limits the replay from being very fast. In fact, it's very similar to rodents, but then there's no choice, but there's something else, something has to give. So what happens is that the replay distance is very short. And so you can think of it as when you have to send information over the internet, right? If you have a small file to send, a very small message, you can transmit it all at once, like a replay, because typically in small environments, the replay it goes through the entire environment. But then if you have a large file to send, then the TCP/IP protocol breaks it into pieces, into packets, and sends in packets. So this might be these packets of information, of short snippets of information that we are seeing. Another interpretation, we talked to people who worked on deep neural networks, and they train, when one network is training another network, again, the classical way is that for this training is to transfer short snippets of information in a random manner, very similar to what we are seeing here. So these people have been telling us this looks very similar to maybe the idea is that the hippocampal network is training the neocortical network, just like people are doing today in deep neural networks. So these are some possible interpretations of what we are seeing here. In any case, it's very different than what you find in small environments, and it's very intriguing. And lastly, we wanted to ask, is there a behavioral relevance for these replays? So for that, we looked at another type of experiment that I'm going to show you a bit later, where in the tunnel, we had now 2 bats flying. So basically, this is a piece of 5 minutes of the behavior when one bat is resting, and then it's flying, and the other bat is flying, and they're meeting in a particular location, and then it gets here, they rest, and then this bat takes off, and they meet in another location. So every time they meet in a different location, right? And then we can ask, during the rest here, does the position of the snippets of replay of this replay is correlated to where you last met the other bat? Because this is a high-speed meeting or collision avoidance, so it's an important event. And so when he's resting here, does he replay the position of the last meeting with the bat, or when he meets here, does he replay this position? So is there correlation between the replay position and the previous crossover position during the behavior? And the answer is yes. This is data from one bat, so it's very preliminary, but but Tamir is now working on crunching the cluster with the decoder for the other bats. But for this one bat that we have so far, there's a clear correlation here between the replay position and the last crossover position. So there seems to be a behavioral relevance for these snippets. And we have now building also, not building, but actually finished building a 700-meter long tunnel. So we demolished the old 200-meter tunnel. We have now a 700-meter long tunnel. This is the tunnel, and a large maze, about 60 by 5 meters in size. And in the tunnel, we are asking, we're already running experiments in this new tunnel. We're asking questions about coding of large spaces across hippocampal subregions, so not just in the hippocampus, but also not just in CA1, sorry, but also in other areas. And we're already doing that, and we're seeing interesting differences there. So all of this is work in progress. We also want to ask, or are asking, about the representation of a multi-scale environment. So this little tunnel here looks like the big tunnel in cross-section, but it's very small, and the big tunnel is big. So the question is basically, you know, I'm sitting here in this room. So this room is small. So I probably have a high resolution representation of this room, but if I go outdoors, my representation of low resolution, right? So the question is, this small tunnel, which is like my room, are the same cells, place cells representing this small tunnel and a large tunnel, or are different neurons? And if it's the same cells, do they rescale their place fields, etc.? So there are many questions like that. And all of this we're doing while recording large ensembles of neurons in flight. And you know, the numbers of neurons keeps growing over the years. As technology improves. And in the maze, we are starting to ask questions about how do we navigate in complex environments, because actually there's never been electrophysiological study in such a complex maze, and we have some specific hypotheses here. Also, multiple goals that the animal has to navigate to, what happens in the brain when we need to reorient ourselves. So when you block the way and the animal needs to sort of recompute and go to another way, what does it mean, reorientation in the brain? The aspects of decision-making, planning, so many cognitive issues involved in real complex navigation, which we are starting to explore in this large maze. Okay, maybe I'll have well, actually we are running late, so I don't know, maybe I should just continue all the way to the end and then ask questions at the end. What do you think?
Probably yes.
Okay, so let me just, sorry, I didn't even note the time. Okay, so I'll just move to a higher gear a little bit. So another direction that I mentioned is about social coding in the bat hippocampus. So this is a work by David Omer, where we asked about the social factors, as I mentioned. And basically the idea, so David Omer now has his own lab at the Hebrew University, but when he was a postdoc in my lab, he asked the following question. Bats are very social animals. And so can you find in their hippocampus, just like you find place cells, which are neurons that encode where I am in space, can you find neurons that encode where you are in space, right? And so basically we designed this task with two bats. I won't go into all the details, but basically there was a demonstrator bat and an observer bat. The demonstrator was flying from the start ball either to ball A. In this case, it flew to ball A, so the observer is supposed to mimic it and go to ball A. If the demonstrator goes to B, the observer also is supposed to go to B. So it's a simple mimicry task, but the idea is that the observer has to pay attention to the position of the other bat. And this is an example, one neuron that classical place coding. You see there are many spikes in a particular location when the bat itself is flying. On the right is when the other bat is flying. So I'm stationary now on this start ball, but still the neuron is encoding the position of the other bat, in this case on the right arm. So in this particular neuron, the social place field was different from the place field. We call these neurons social place cells because they encode the position of another animal. In other neurons, the self place field and the social place field overlap. So there was a heterogeneity there. And we ruled out trajectory planning as explaining these results, because you might think maybe I'm not really representing the position of the other bat, maybe I'm planning my own trajectory, so we ruled that out. Also, we asked, is this really social? What happens if you move plastic objects, right? Maybe anything is being encoded. So the answer is that some of these neurons also encoded an object out there, but it was always different from the conspecific, from the social response. So there's something special about the social response. And so we think that social place cells may be important for social spatial cognition, for representing conspecifics, predators, prey, important things out there. In particular, for example, we are as humans are very good at this. Think about basketball or football players. What they're really good at, the good players, is they always know where their teammates are, the other teammates are. They really track their positions. So maybe these neurons are what's doing that. And we've now moved with Psychiatry, my postdoc moved to a even much more crazy experiment where completely uncontrolled situation, but still very interesting, where he has asked, because what I've showed you here is representation of one other individual, but the question is what happens when you have multiple other individuals. So Psychiat set up this room where you have between 5 and 10 bats, both males and females, and we developed methods to track the identities, positions, and head directions of 10 bats simultaneously in this three-dimensional room, which is not trivial, but we solved this. And then we can record from the brains of 1 or 2 or more animals simultaneously while they behave here. And now there is no task here. They just live here and interact. And you might say it's completely uncontrolled. But on the other hand, which is true, on the other hand, as I said in the beginning, they do repeat certain behaviors over and over again. They do, fight over and over, or have sex, or they, feed each other, or there are many social behaviors that repeat many times. So you can actually build rasters and look at that. So it's not as uncontrolled as it seems. And just to give you a flavor, again, this is ongoing work, but to give you a flavor of what are we seeing, this is an example now in flight. This is when you're flying between two corners of the room, this is a neuron that has a place field in this location. So you'd say, well, it's just a regular place cell. But when you look at the same neuron, this is when it's flying, when there are other bats at its destination. But when the destination is empty, actually this neuron almost doesn't fire, right? So it's not a regular place cell, but it's actually very strongly modulated by a social factor, right? Am I going to a party or am I going to an empty party, right? This neuron cares a lot about this. We also found that some of these neurons encode identity of the bat. I won't go into the detail in here. And we also looked at what happens on the nets locally when they're interacting with each other, not flying. Now, this is a difficult problem because if you think about it, it's a very high-dimensional problem. You have here the recorded bat. So there is the position of and the head direction of the recorded bat. Then there are the positions of 5 other bats, the distances of 5 other bats, angles to 5 other bats. It's easily a 20-dimensional behavioral space. It's not your one-dimensional tuning curve of Hubel and Wiesel of orientation, or even not a 3-dimensional place cell. It's a very high-dimensional behavioral space, maybe something like 20 dimensions. So how do we analyze this? So we used generalized additive models. It's like GLM, only the GLM is linear and general additive models are non-linear, so they're more powerful. But on the other hand, they are they do give you explainability, meaning you can ask which of the factors contributes to the model. So here I'm showing you just an example of one neuron. In blue is the firing of the neuron over 1 hour or so, and in red is the predicted firing of the model. And you see that it tracks the neurons firing very nicely with a correlation of 0.6, much larger than for the shuffles. But then you can ask the model which factors contributed to the firing of this neuron. It turns out for this neuron, there were 3 factors. One is the head direction. This is the actual empirical head direction tuning curve of this neuron. And this is the tuning predicted by the model. So very nice overlap. This neuron also cared about the position of the recorded bat. So it's a classical place cell, you might think, right? This is the actual data and this is the model. But the surprising part is this neuron also said, the model says that this neuron also cares about the position of bat number 5, right? Is not bat number 4, not bat number 3, not bat number 2, just bat number 5, right? So there's an identity coding here as well. So these, and we see that some neurons, we have hundreds of neurons. Some neurons encode just my own positions, or are classical place cells, low dimensional in the sense that they encode just a few and a small number of behavioral dimensions. Other neurons are 10-dimensional, can encode 10 different behavioral variables significantly. So we have a whole zoo here, and we're analyzing this, but it's all very interesting. And clearly, they're very strong social effects here, very strong social encoding in the hippocampus, and very clear identity coding as well. Okay, so this was briefly about the social stories, and the last story that I hope I won't run overboard over time, but let me run through it quickly, and that's my last story, is about the issue of natural switches in behavior. This is work by a PhD student Eretz Sarel, together with PhD students Shaked Palgi and Dan Bloom. And so the rationale, or the basic question, is the following. Behavior is very dynamic, right? So when I live a few minutes walk from the lab, from campus. So when I walk from my home to the lab, it's really a few minutes. Initially, I navigate. Then I get to a junction. I need to decide, do I go to the right or left? Because I have two ways to get to the lab. Then I might bump into somebody, a colleague, and chat to them. So social interaction. So within just a few minutes of getting from home to work, I switch from navigation to decision-making to social interactions, right? This is real life behavior. It's very dynamic, things change. But that's not how we do neuroscience. In neuroscience experiments, we typically study one thing at a time. You study just navigation, and another lab will study just decision-making in a different brain area. And a third lab will study social neuroscience in a different country, in a different brain area. So it's very segregated, but that's not how real life behavior works. And we wanted to understand, when behavior is very dynamic, what happens in the brain. And particularly, navigation is very dynamic. When an animal navigates, it also needs to pay attention to things out there, like a conspecific or a predator that may attack it, etc. So the question is, do hippocampal neurons rapidly switch to represent these other things out there, and what happens? So specifically, what we did is this two-bat experiment in the tunnel that I already described, and where we had two situations in the tunnel. Either one bat was flying or two bats were flying and crossing over each other. And this were not two different sessions. This was completely intermingled, right? The idea is that when this bat is flying, is resting, and this bat is flying, this is solo, but then in the next trial, they might fly and cross over, right? So this is completely intermingled. And as you saw already, they meet each other in different locations in the tunnel. And so what might you expect? If these are like two hypothetical possibilities, we might, if it's a classical place cell, then if we plot the firing rate of the neuron, red is high firing rate versus position and interbat distance, we expect to see something like that, right? This is like a classical place cell that has one place field. Of course, if it has several place fields, it'll have several of these bands, but they will be horizontal. There will be no modulation as function of distance if this is a classical place cell. On the other hand, if this is a distance coding cell, you might expect to see something like that, let's say distance 0 or some other distance. And indeed, we found some neurons that look like that. So here is an example. This is now a real neuron that has these horizontal bands, more or less only cares about position. And this is a neuron that more or less cares about distance, not so much about position. And so here is its distance tuning. But most neurons were more complex than that. For example, here's a neuron that only fired at a particular combination of distance and position. And here's a neuron that was actually inhibited at a particular distance from the other bat. So we saw all sorts of responses there. These neurons, these responses could not be explained by changes in speed or lateral deviation or anything like that. We had controls from that. This is not published yet, but it's actually been a few days ago been accepted for publication. So you'll see this out very soon. And if you look at the population of the neurons, so if you now look at basically at these one-dimensional tuning curves, and you color code them and look at all the neurons. So now each row here is a color-coded distance tuning. You see that you have all possible distances, but clearly you have an overrepresentation of distance 0, of the short distances. Now, interestingly, here we can utilize an interesting property of the sonar, right? I mentioned the sonar. One thing we know already for many years, and it's been shown in many bat species, that when a bat attends to something, it increases the rate of its echolocation calls. And you actually see it here. This is a raster, not of spikes, but of echolocation calls. Each dot here is an echolocation call or a sonar call. In here, 52 trials are 52 of these crossovers. And you see that when the bats gets close to each other, so, 15 or 20 meters before they meet each other, they start to increase the rate of echolocation call, and then it goes down after they meet each other, right? So there is essentially an index of attention. We can look at the rate of the echolocation calls or the click rate, and this gives us an index of attention. This is amazing because if you are looking at a rodent, a rat or a mouse that is freely running around, there's no easy way to measure when is it attending to something. It's very hard to measure. In the bat, we just put a microphone and we have here an index of its attention. And it turns out that this attention, spatial attention, modulates spatial representation in the hippocampus. This is something that Shaked Palgi has shown. He separated the flights into flights with high click rate or high attention versus flights with low click rate or low attention. And he shows that here are two example neurons, that the firing rate increases is higher for the high attention flights. And this is just the examples. And here is the population. It holds across the population. And this cannot be explained by auditory responses, right? Because of course, if this was a neuron in auditory cortex that responds to an individual echolocation call, or maybe a motor neuron that responds to an individual echolocation call, now of course, when your rate of echolocation increases, you have a higher firing rate. But we actually did this. We did a echolocation call triggered firing rate, a histogram or PSTH, and for the vast majority of the neurons, they are flat. So these are not auditory neurons. Okay, it's the hippocampus. You don't expect them to be auditory neurons, but they are not. And so it's not really an auditory response, but it is a state of high attention versus state of low attention. Okay, so attention modulates the spatial coding. And lastly, we looked at how does the distance coding changes as a function of position. So here are examples of two neurons on the left and on the right. So in this neuron, at this position, this place field, it was excited at a particular distance. But at another position here, it did not respond at all, didn't care about distance, right? You see it here in the map. It's tuned to distance here and it's flat, doesn't care about distance there. So different distance tuning at different locations. This neuron has an excitation in one location, but an inhibition in another location. So we analyze this by looking at correlation between place fields of the same neuron, of the distance tuning field, sorry, of the same neuron in different place fields of the same neuron, in pink versus different place fields of different neurons. And you see there's almost perfect overlap. There is some overrepresentation here on the right, but it's almost completely overlapped, which means that different place fields of the same neuron have almost independent distance tuning functions. So very surprising. And we also did some singular value decomposition and other analysis to show that indeed these maps are non-separable. When I say non-separable, I mean that this is a two-dimensional function. It's a function of x and y, of distance and position, and it cannot be actually explained as a multiplication of the marginals of a function of x and a function of y. All right, it's really non-separable. You see here in this previous example, these two-dimensional functions can be pretty crazy. And again, we teamed with Yonatan Al-Khadef, and he did a theoretical decoding analysis to show that such a non-separable representation improves coding. Again, I don't have time to go into the details, but I can explain more later. So to summarize this last part, during crossovers, many place cells in the hippocampal area CO1 switched from coding of position to coding of distance by position. This switch was very fast. I didn't have time to show you, as fast as 100 milliseconds, very, very fast. So when the bat is flying alone, it's just encoding its own position, but when it approaches the other bat, within 100 milliseconds, there is a switch to representing my own position, but also the distance to the other bat. Again, here we're utilizing the fast flight speed of the bats. Because they fly so fast, the behavioral switch is very fast, and then the neural switch is very fast. And these results suggest that neural circuits can modulate very rapidly the core computation they perform. Neural coding can be very dynamic. We typically think about the function of a neuron as a given, right? This neuron does that. It encodes orientation in V1, or it encodes place or whatever. Here, the neuron switches from doing one thing, encoding of position, to doing another thing, which is coding distance by position. So this demonstrates that the neural code can be very dynamic. So let me summarize. These are now my last two slides. To summarize overall what I showed you today, we discovered novel types of neural coding when incorporating natural aspects of behavior, right? None of this was really completely natural, but we did a step into a more natural direction. And every time we did a step into a more natural direction, we found something interesting, right? We went to 3D and we found 3D plays, 3D maps, 3D compasses that were toroidal, and the 3D grid cells look completely different. We killed most of the models of grid cells. We went to large natural spatial scales, all hell breaks loose. The coding looks completely different, this multi-scale, multi-field code that I showed you. And we also have a theoretical understanding why it's good. We introduced additional animals, we allowed social interaction, and then we found representation of other animals. We allowed the animal to switch between navigation and other behaviors, and then we find this very dynamic and rapid switches in the neural code. So every time we go in the direction of a more natural behavior, we'll find something interesting. And so my overall take-home message is the following. It's about the importance of natural behaviors versus the classical reductionist approach. So as you know, in the last 400 years, since Galileo Galilei and Descartes and Francis Bacon, science has been very reductionist. And I completely agree that reductionism and control are very important. But on the other hand, I worry that if we are trying to understand behavior, we might be shooting ourselves in the leg, because if we control too much, if we reduce too much, then we are missing the thing that we came to understand, which is behavior, because behavior is rich, is complex, is context dependent. So if we simplify too much, we might be killing what we're trying to understand. And so I think, therefore, it's important to look also at more natural behaviors, not instead of reductionism, but in addition to the reductionist approach. I don't think it's healthy for neuroscience that 99.9% of the field is doing very reductionist and very ultra-controlled experiments. I think there should be more people going to more natural behaviors. And as I showed you, every time we went into a more natural behavior, we found something surprising about the brain. And I'm writing now a book for MIT Press called Natural Neuroscience, where I hope to convince the neuroscience community about the importance of studying the brain closer to natural behavior of the animals. It's not, this book is not about bats and not about even navigation. The whole of neurobiology of navigation is just one out of 8 chapters, but it's much broader and tries to make the argument that in every sense, every system, in active sensing, in sensory neuroscience, in navigation, memory research, and decision-making, in social neuroscience, there's already hard evidence out there showing that the brain cares, neurons care about whether the behavior is natural or not. So these things are very important. And so I'd like to finish by thanking people in my lab, especially Liora Lass, who is a fantastic staff scientist working with me. She's been involved in almost everything I showed you today. And I think I mentioned all the other people in the lab as we went along, and alumni and funding sources. And thank you for listening.